Hello, everyone, and welcome to today's live broadcast, Simultaneously Multi-Analyte Profiling of Rare Antigen-Specific T Cells with 3D Flow Analysis, presented by Dr. Karen Anderson, an Associate Professor at Arizona State University, and Dr. Doug Heinerfeld, a Principal Product Application Scientist at NanoString. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by NanoString. NanoString Technologies is a provider of life science tools for translational research and molecular diagnostics. The company's molecular barcoding technology enables a wide variety of basic research, translational medicine, and in vitro diagnostics application. Before we begin, I want to remind everyone that today's event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on that green Q&A button located at the lower left of your presentation window, type your question into the box that appears on the screen, and we'll answer as many questions as we have time for at the end of the presentation. Also notice that you will be viewing this presentation in a slide window. To enlarge that window, just click on the screen icon located on the lower right. If you have any trouble seeing or hearing that presentation, use the support button at the top right or that Q&A button and let us know you're having a problem. Good news, this presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left-hand corner and to follow the process to obtain your credits. Without further ado, please join me in welcoming Dr. Karen Anderson and Dr. Doug Heinerfeld. I will now turn the presentation over to our speakers. Welcome, both of you.
Good. Thank you. This is Doug Heinerfeld, and as you just heard, I'm a Principal Product Application Scientist at NanoString, where I focus primarily on the applications of our 3D biology assays, looking at DNA, RNA, and protein, like you see here on the screen, and I'll tell you some more about that. So NanoString has a long track record in analyzing RNA, but certainly we know and you know that RNA does not really explain the full complexity of cell biology. So recognizing that, we've moved to adapt our technology to look at a broader range of analytes. So just as a simple example of why it's important to look at both RNA and protein, in this study where we've stimulated macrophages, and we've done a time course here, we're monitoring both RNA and protein. So we can see here for CD4, after you stimulate the cells, you see an increase in both the RNA and the protein, and then a concordant downregulation of both, which contrasts to CD9, for example, where the RNA and protein are both increased after stimulation, but then the RNA falls off, but the protein, which we're measuring at the cell surface, remains strongly expressed. So there's clearly post-transcriptional regulation that's going on either in the translation or the transport of CD19, or CD9 rather. So just as another example of looking at both RNA and protein, this is a paper that was published in 2016 looking at HER2 resistance in breast cancer. And they took an approach using publicly available data where they looked at data from TCGA, both reverse phase protein array, data looking at, in this case, phosphorylation of YAP, which is part of the Hippo tumor suppressor pathway. And then they looked at that phosphorylation, and they looked at genes whose expression either correlated or was anti-correlated with phosphorylation of YAP1. So in that way, they could learn a lot more about the biology by both looking at protein phosphorylation and gene expression at the same time. So just as another example, this was a fairly recent publication from GSK, I believe, looking at both targeted therapeutics and immune modulators such as PD-1, PD-L1, CTLA-4. So in this study, in order to understand more about the molecular mechanisms of these combination therapies, they did both RNA analysis by NanoString, but then they did extensive protein analysis using flow cytometry, immunohistochemistry, Western blots. And it was really these kinds of studies that inspired us to create a platform where we could look at all these different analytes at one time. So doing that, of course, requires multiple, traditionally I should say, requires multiple technological approaches, maybe high throughput sequencing, mass spectrometry, cytogenetics, so imaging type technologies. Studies like that are time consuming and often quite expensive. They also generate data sets that are quite disparate in their structure, so it's very hard to integrate those data. They're oftentimes filled with lots of noise. You typically need a team of bioinformaticians and statisticians to try to pull that data together. But I think even more importantly than those two items is the fact that oftentimes you just don't have enough sample to look at RNA, protein, and DNA, for example, from a very small amount of sample. So just to tell you a little bit about our platform, so on the left here you see the nCounter platform, which is our technology that we use to analyze and count different molecules within the cell. So we can look at protein, we can look at single nucleotide variants, we can of course do gene expression, which again has been our bread and butter for many years, gene fusions, copy number variation, and we have applications where you can go from single cells. So one of the key features of this is from a single small sample, we can analyze up to 800 different targets. So those targets can be, again, combinations of DNA, RNA, and protein, but the total number of analytes is 800. And we do this by counting molecules. The way we do that, we have two parts to our nanostring barcode chemistry. One is the barcode itself, or the reporter probe. So this is 4 different colors and 6 different positions. So by changing the combinations of those, we can create unique barcodes. And then we have a capture probe here. So if, for example, we're analyzing RNA, we take those probes, we mix them with the RNA. The RNA then binds to the specific target sequences that are designed here and here. And when this complex comes together, we can then count the numbers of times that RNA is present in the sample. You'll notice I didn't say anything like reverse transcription or PCR. So in these applications, you're directly counting the molecules from the lice sample. So we image those barcodes on the system as shown here. So you can see different barcodes showing up with different series of colors. Each series of colors represents an individual molecule that we're counting. One of the beauties of nanostring is that the data is very simple. So here we have different sample types across the top, and across the side we have different analytes, and then we're just simply counting those. So we see whole number counts for each of those analytes, and then we can ask whether that RNA, protein, DNA molecule is differentially present between the different samples just by a simple counting exercise. So again, you saw there that with RNA, it's quite straightforward. We have the barcode and the capture probe, and we hybridize it to an RNA. Now those sequences here and here are designed to be specific to the RNA of interest. For DNA, which we're not going to talk about in great detail today, but we have applications around, particularly around solid tumors, where we look at DNA. But really, the whole action is right here. So if we have a perfect match between that sequence and the DNA that we're interrogating, then this complex is stable and you get a digital count. If there's a mismatch in that region that I highlight, then that complex is unstable and you don't get a count. So we design a probe to the reference wild type allele. We design a probe for the specific mutation that you're looking at analyzing, and then we compare the counts between those. And with that technology, we're able to identify single nucleotide variants or small indels. On the protein side, what we've done is we've taken antibodies. So these are antibodies that we purchase from vendors that are well used. We then conjugate to them a specific DNA barcode shown here. So if the antibody binds to the sample, we have a unique barcode, and then we actually interrogate that barcode with our reporter probe and our capture probe. So we're counting these DNA molecules here. If that DNA molecule exists, we know the antibody bound to the sample. We know the protein was expressed. So this is all an integrated platform where we can look at DNA, RNA, and protein all together at one time and get digital counts for all those molecules on the nCounter system. The inputs are very low. So for DNA, it's 5 nanograms of DNA. For RNA, it's 25 nanograms. if you're purifying it, or 2,500 cells if you're just lysing the cells. Again, you don't have to purify the RNA. You don't have to do reverse transcription. For protein, so it's, depending on the assay, 250 nanograms, or as it says here, down to about 1,000 cells required to look at the protein analysis. So this 3D biology concept, where we're looking at RNA and DNA and protein, is really built on the backbone of these pan-cancer 770 gene hallmarks of cancer gene expression series. So these have been out for some time. These are off-the-shelf panels that we provide with 770 genes. We have on the left the pan-cancer pathways, which really focuses on how the tumor starts. We have content related to DNA damage and PI3 kinase and MAP kinase and that sort of content in there. In the middle, we have the pan-cancer immune profiling, which is really about the immune microenvironment and how the body is actually responding to the tumor. And then tumor progression, things like EMT and ECM and metastasis and so forth. So these panels have been available for some time and continue to be available. So another panel that I'm going to talk about is the IO360 panel. But in order to talk about that, I first want to bring up something called the tumor inflammation signature. So this is an 18-gene panel that looks at different aspects of immune biology, interferon gamma biology, T-cell exhaustion, and so forth. These genes have all been known to be related to response to PD-1, PD-L1 inhibitor pathway blockades. We have an algorithm where we can take the digital counts from these different genes and determine whether that tumor is in fact immunologically hot or cold, and the degree to which that is true. And as you'll see here, I'm sorry it's a little bit blurry here, but this paper was recently published by Merck, where they looked at interferon gamma related mRNA profile predicts clinical response to PD-1 blockade. So it's a great paper. There's a lot of data in there. This particular figure just highlights a comparison of looking at PD-L1 by IHC versus this 18-gene tumor inflammation signature, and shows an increased area under the curve with the 18-gene panel versus the PD-L1 IHC. So improved performance, and this was in head and neck cancer. And again, this has been published. So that tumor inflammation signature is one part of a larger gene panel that we've developed called IO360 that looks at the tumor, looks at the microenvironment, and looks at the immune response. It's a panel that's really commonly used in FFP samples, so it's an RNA panel. So it's enriched for known and potentially clinically relevant genes. It is compatible with the protein, with either adding additional RNA or using our Vantage 3D protein panels. I'm not going to talk about the panels today related to this, which are things related to targeted pathways like PI3 kinase, but it's a great panel to look at immunological activity within tumors. To go one level deeper into that, this is another way you can look at the data. So if you take an FFP sample and purify RNA and analyze it on this sample, the first thing, using this tumor inflammation signature, you can tell whether the tumor is hot or cold. And then if it is in fact hot, you can then look at, for instance, PD-1, PD-L1 pathway activation only, which would indicate that the PD-1, PD-L1 blockade might be a good option. It may be suppressed, so then you need to activate other pathways. So you can see that here in the inflamed tumor state. And then if it's cold, you can look at, it has genes that help you understand whether there's defects in T-cell trafficking or priming. And then as you see here, it goes on to then give you information that would help you select other types of therapeutic approaches on that tumor. So a lot of information about therapeutic strategies that would be used for tumor. So I'm going to jump from that and kind of then bring it together where we're looking at both RNA and protein. So these are our Vantage 3D assays. So in this one in particular, looking at the immune cell profiling and signaling assays. So again, we're looking at RNA and protein together. The panels that we have available today include 770 RNAs, and that's that pan-cancer immune profiling panel that you saw before. And then you can look at that in combination with either 30 cell surface markers or 26 intracellular markers. you'll see our typical input is about 20,000 cells, but you'll see as we get into some of the specific applications here that you can go down to lower numbers of cells. And importantly, you can look at the content and decide that some of it looks great, but you really want to add some additional content to it. That's no problem. You can send us an antibody. We'll add a custom DNA tag to it, send it back to you. You then spike it into the panel. So you can add custom content to the assay. Speaking of content, again, this is the RNA panel here. And then we have either the immune cell profiling or the immune cell signaling. And you'll see a lot of the players you would expect to see, particularly in the space of IO, PD-1, PD-L1, CTLA-4, and so forth. And then down here, these are intracellular proteins. So the way the standard assay works is very simple. So you take your cells in suspension, spin those down and collect them. You take some of the cells out and just lyse them, and that becomes the input for the RNA analysis. Again, no RNA purification. You just lyse them. And then if you're doing the immune cell signaling, you fix and perm the cells. Immune cell profiling, you just stain the cells, the live cells, with the antibodies. Again, our antibodies have the DNA tag. So after you've stained them, washed them thoroughly to remove any nonspecific binding, and then lyse those cells, these tags are released from the antibodies. You mix that with the RNA, hybridize it to our probes, and just count the numbers of RNA and protein expressed in your sample. So we could talk all day about validation, but I'm just going to show a little bit of data here. And this is stimulating cells over a time course with PMA. And in this case, we're looking at PD-L1 and showing changes in a sample that's been either stimulated, treated, or untreated, and showing an increase in fluorescence here with the treated sample, as you would expect. So just another way to look at that here is mean fluorescence intensity, and showing with the treated sample the increase in expression, and the untreated, no increase in expression. So here we're looking at more markers than just PD-L1. We're looking at 3 other markers, and you can see that comparing the mean fluorescence intensity to the digital counting from our NanoString 3D assay, you get very much the same response. But importantly, you're getting the RNA data as well. And this is where you really start to see some interesting results, right? Another example where the RNA goes up along with the protein, but then the RNA is stabilized or even going down, and the protein continues to be expressed at a higher level on the cell surface. So seeing some discordance and really showing the value of looking at both RNA and protein. One of the applications of this has been in adoptive T-cell therapy, of which CAR T-cell therapy is one of the most common. And it's an area where there is a great need to identify biomarkers to understand efficacy and reduce toxicity. So one of the areas in an early study we've done with a leading company in this space is looking particularly at biomarkers to characterize T-cell function and production. So you've taken the patient sample, you've purified the T cells, you've engineered the T cells to express a chimeric antigen receptor, and then before you put those back into the patient, it sure would be nice to know whether that T cell is going to be a highly functioning T cell and is actually going to have the targeted cytotoxic activity that you are hoping for. So the way that these experiments were done was taking the CAR T cell and then incubating that, co-incubating the CAR T cell with a cell that either expresses the specific target for the CAR or a non-specific target. And so in this case, we hope with the specific target, we hope to see a strong cytotoxic activity of the CAR T cells, and then not with the non-specific target. And essentially that's what we see here. We're looking at 770 RNAs and 30 proteins using our immune cell profiling. And we have the CAR T cells with the specific target or the non-specific target. And we can see very strong differences in the expression, which will help some collaborators here learn more about the biology of their CAR T cells with respect to they're hitting their specific target. But really what they want to know is they want to know, how does this relate to clinical outcome? And so just in a very small pilot study, we've started to look at that. So same starting data set, RNA and protein, and comparing those patients, samples from the patients that were non-responders versus responders, and starting to see a difference. So these are statistically differentially expressed RNAs and proteins in the assay between the non-responders and the responders. Now, if you recall, the assay going in is 30 proteins and 770 RNAs, but what we actually see is that there's 5 proteins and 22 RNAs that start to make up a sort of a candidate signature that that our collaborator is going to go on to continue to pursue and refine towards clinical application. A really great example of using this 3D biology technology, in this case starting with a partially purified clinical sample, going right into our assay, and then seeing changes in gene and protein expression. We also pretty quickly realized from talking to customers that there was an interest in integrating that with flow cytometry to look at very specific populations of cells. So the idea being, and we call this 3D flow, the idea being is that understanding the function of specific immune cell populations in disease or therapeutic response requires sophisticated analytical methods, and that those methods are complex, oftentimes inefficient and costly. So you might be combining with high parameter analytical cytometry or cytoph analysis with sorting specific populations of cells and doing nanostring or qPCR or RNA-seq. So you're using multiple technologies to look at these. And again, oftentimes you don't necessarily have a lot of sample, but you know that the target population of cells you want to look at, which might be quite rare. So 3D flow expands on existing flow cytometry methods to enable a deep analysis of defined cell populations, again with the goal to discover biomarkers or therapeutic targets for IO, autoimmune disease, or any area in which the immune cell is of particular interest. So the way this assay works is you know the cell populations you want to go after. So maybe you want to go after Tregs, or you want to go after, as we'll hear, antigen-specific T cells. So you stain the cell with standard flow cytometry antibodies that are fluorescently labeled to sort the populations of interest. But at the same time, you're staining those cells with our DNA-labeled antibody cocktail, which has 30 cell surface proteins. You then sort the cells based on the fluorescent markers, lyse the cells like I've described, which releases the RNA and the protein tags, and they hybridize together, and then you're able to characterize the RNA and protein from those populations of cells of which you are interested. So our content from the protein side, this is another look at it, we looked at it before, has a lot of excellent content for T cell profiling. So it has content related to sort of general T cell biology, activation, inhibition, and so you'll see some more information there. And of course, all this information, the content, the clones, everything is available, so you can peruse through that. And again, if you see if there's anything missing, we can add content to these, so we can add custom content. Again, just another look at it here. We're starting with a larger T cell population. We're then sorting specific T cells. They're stained with our DNA-labeled antibodies, and we're able to characterize the expression of all these antibodies in those rare populations of cells. So you might be asking about the underlying biology of these cells, either in an exhausted state or an activated state, how those T cells change before and after immunotherapy. So you may be engaging a target on a T cell, and you may or may not see response in the patient. So if you really want to understand what's going on with that T cell, when you have response in a patient or lack of response, this is a great approach to dig right in and interrogate those T cells. So this is, again, how the system works. You stain the cells with the fluorescent markers. We see the fluorescence here with the CD3 positive T cells, the CD19 positive B cells, and then the CD8s and CD3s. But each one of these cells, each one of these spots is also coated with DNA-labeled antibodies, which at this point you don't see. But if we sort these 4 different populations, and we sort either 5,000 of those cells or 500 of those cells, we get data that looks like this. So each one of these spots is the normalized digital counts for one of the 30 proteins in our assay. So we sort either 5,000 cells or 500 cells, and what we see is we get a very high correlation. And what this tells us is that the assay is clearly sensitive down to 500 cells for protein analysis specifically. And we repeat this with CD4, CD8, CD3, and the CD19 positive B cells. So very reproducible, very sensitive for very small numbers of cells. And again, this is looking at those 30 protein markers alone. So another way to look at that is here we have our CD4s at the different populations, CD3s, CD8s, and CD19s. And this has been a heat map showing the expression of all the proteins in our panel. And so you can see, for example, here we have CD19. So there's a DNA-labeled antibody there, and there is the fluorescent labeled antibody that was used to sort the cells. And we see very strong expression in the B cells and almost no expression in the T cells. Same sort of result, here we have CD3, where CD3 is expressed in all the T cell populations and not in the B cell population. So it's a great way to get a lot of information about very small numbers of cells. So that was protein alone. We have, of course, looked at RNA as well. And so in this experiment, we were sorting CD4 positive or CD8 positive T cells. We sorted either 5,000 or 15,000 cells, and we did triplicate conditions for each. So this just shows using principal component analysis, which is a great way to look at, what explains the variance in the data. It could be 15,000 versus 5,000 cells, for example, but it's not. It is CD4 versus CD8 that explains the majority of the variance there. So you can also look at it in a heat map where you're looking at both proteins and RNAs, but this is looking at 770 genes and 30 proteins all at the same time. Kind of hard to make much sense of that. But one of the nice features of our system is we also have a software called Insolver, and using the advanced analysis component of that software, you're able to take the data and analyze it in a lot more depth. So you can take all the RNA and protein and then just limit your analysis to genes and proteins related to T cell function. So that's an example here. And you can see, again, here's the protein expression changing down here between the CD4s and CD8s. This is an unbiased clustering. So again, we see the CD4 cells clustering separately than the CD8. Very strong differences in that clustering. You can go in and look at the TNF superfamily. So there's a whole variety of different sort of functional gene sets and protein sets that you can interrogate within these cell populations. Again, here we see, although we start to see an intermixing of the 5,000 and 15,000 cells, we see clearly, again, the CD4s and the CD8 separating in a very, very strong protein response down here. So taking those cells, this is another component of our advanced analysis. And here what we're looking at is the correlation of RNA on the y-axis and protein on the x-axis, or in this case, ITOS. So what we see is in the CD4 cells, we see an increase in both RNA and protein expression. So again, we're looking at RNA from the lyse cells. We're looking at protein expression from protein expression on the cell surface. Again, for CD4, as you would expect, it has increased protein, but also has increased RNA. And then in T5E is an example where there's no increase in the RNA expression between the CD4s and CD8s, but there's a huge increase in protein expression. So again, looking at RNA or protein alone really doesn't give you a full picture of the biology and the regulatory mechanisms going on with these cells. Looking at both at the same time from even very rare populations of cells, you're able to really understand the regulatory mechanisms going on here. So we've also done a number of other experiments. The populations of cell types that we're looking at continues to grow, but this is a very simple experiment just looking at protein and RNA analysis from 5,000 either stimulated or unstimulated T cells. And this is a volcano plot here where we have fold change left to right on the x-axis, and then we have p-value on the left axis, and it's the inverse log of that. And so these sorts of, we see genes and proteins here as well. So we have the protein and the genes. And so these molecules are very strongly differentially expressed from both a log change, a fold change, and a p-value, and then we see those that are increased expression with stimulation. So a really great example of how much data you can get, how you can quickly use the software, in fact, in the analysis approach to identify genes and proteins that are differentially expressed from very rare populations of cells. So I just wanted to bring up this one additional panel that we're in the process of developing. And so this is a panel focused on autoimmune disease, and I wanted to show it because I thought it might pair quite well with the protein assays, particularly if you're doing research in autoimmunity. So this is, again, a panel that's going to be something on the order of 770 genes. We're developing in collaboration with Institut Pasteur. And the way that the initial approach to creating the gene set was actually to take all of these different autoimmune diseases shown over here, you really can't see it in the plot there, and look across all the GWAS hits and pick genes associated with those GWAS hits. So in that way, we're getting genes that are known genetically to associate with the disease state. And then on top of that, we've added additional genes sort of more related to just general immune response genes and immune cell function. So I think this is going to be a great panel to couple with the protein panels that we have for your research into autoimmune disease. And with that, I'm going to turn it over to Dr. Karen Anderson, who's going to talk to us about the approaches she's taking with this to look at antigen-specific T cells. Karen, it's all yours.
Thank you so much, Doug. So what we have been interested in is doing deeper immunoprofiling of antigen-specific T cells, in particular looking at markers of exhaustion along the continuum of T cell exhaustion, especially in the setting of chronic antigenic stimulation with either tumor or with viruses. And with that, let's see if this works. There have been a number of studies over the years looking at antigen-specific exhaustion and exhaustion biomarkers. This was a classic study out of the laboratory of Rafi Ahmed looking at chronic LCMV infection. And if you look at this in the murine model of LCMV infection, you can see the induction of exhaustion, and with exhaustion of antigen-specific T cells, you can see upregulation of a number of different inhibitory molecules, both at the RNA level and as well as at the protein level, and downregulation of signaling components for intracellular signaling components, and ultimately with loss of cytokine secretion with interferon gamma or with TNF alpha as shown here. So under chronic exhaustion of antigen-specific T cells at day 30, you can see loss of interferon gamma and TNF alpha secretion. You also have loss of self-renewal and proliferative signals as well. And this happens more specifically under chronic antigenic stimulation, whereas acute infection, you have maintenance of both cytokine secretion and proliferative potential in these cells. And so identifying some of the molecular mechanisms underlying this has been efforts of a number of different laboratories and a tremendous amount of research over the years. In this study coming out from the Ludwig Institute in Lausanne has been looking at chronic activation states of antigen-specific T cell exhaustion, and in particular looking at tumor-specific T cell exhaustion in the peripheral blood versus tumor-infiltrating lymph nodes and tumor-infiltrating antigen-specific T cells, in particular for melanoma and melanase-specific T cells. And so in this study, if you compare Epstein-Barr virus-specific antigen-specific T cells to chronic CMV antigen-specific T cells to melanase-specific T cells, what you can see is upregulation of a number of different checkpoint inhibitors, but what you even see specific differences in the blood versus infiltrating tumor-infiltrating lymph nodes of melanase-specific antigen-specific T cells, both with, in particular with CTLA-4 and also with TIM-3 and other biomarkers of exhaustion, and also biomarkers of activation. And so there is this difference and this need really to identify deeper profiling of the level of antigen-specific T cell exhaustion, and that it may vary between different sites within people as well as between different antigens. And so as these CD8 positive T cells certainly are undergoing exhaustion, they lose effector functionality, and it appears to be a stepwise fashion. If you're looking at antigen-specific effector functions versus memory cells versus exhausted cells, you can see stepwise loss of interferon gamma or TNF-alpha expression in chronic infection and also in the development of this almost anergy-like state and this difficulty and impairment of cytokine signaling in these patients. And so what this ends up leading to is that there is this, this progression along this exhaustion continuum of antigen-specific T cells, certainly for CD8 positive T cells. And in this very nice review published about 2 years ago, looks at a number of different and a progressive loss of different biomarkers of exhaustion, including cytokine secretion, but also loss and fairly early loss of cytotoxicity and the progressive increase in expression of inhibitory receptors and the loss of proliferative potential in these antigen-specific T cells. And so we were interested in particular at looking at a number of these different biomarkers and to do more deep profiling of antigen-specific T cells, but to be able to do it in a more rapid way rather than doing isolating for RNA-seq analysis or other types of approaches. So we were interested in sorting antigen-specific T cells and looking both at protein profiling and RNA profiling. And so what the challenge is, and the fundamental challenge with many of these studies is just simply the limitation in the numbers of T cells. And in this case, we are looking at pre-stimulation frequencies are often less than 0.1% of CD8 positive T cells. So these are very low frequency events within isolated peripheral blood mononuclear cells. And so we were interested in having sensitive ways of being able to look simultaneously at both RNA and protein expression. And so what we did was look at antigen-specific T cells that were specific to the influenza M1 protein, in particular a peptide derived from influenza M1 restricted to HLA-A2. And so we looked at peripheral blood of healthy donors and looked at both CD8 positive, tetramer negative, and CD8 positive, tetramer positive populations that underwent FACS-specific flow sorting and then nanostring-based immunoprofiling both on the protein level and on the RNA level. And to do this, we were actually measuring down to about 400 CD8 positive T cells in each population. And in this case, these tetramer positive populations, well, both populations really underwent in vitro stimulation for a short term in order to increase the pre-sort frequency of the antigen-specific T cells. And so as you can see, there was an increased relative amount of 4-1BB, CD27, and a number of other co-stimulatory, co-inhibitory molecules, as you would expect, in stimulated antigen-specific T cells relative to stimulated but antigen-negative T cells, CD8 positive T cells that were not stimulated with specific peptide. And in comparison, looking at the RNA profile also obtained from approximately the same number of antigen-specific T cells, you can see elevated levels, for example, of granzyme B or perforin relative to the non-specific CD8 positive population. And so we can start to do more deep levels of profiling of these antigen-specific T cells from peripheral blood and potentially from other sites. And currently we are looking at the differences between different types of antigen-specific T cells, in particular influenza versus chronic viruses such as CMV or EBV within human populations, and then ultimately to more tumor-specific, chronically antigenically stimulated T cell populations. And I think that tools such as these provide the opportunity to do that on a more rapid integrated platform system to be able to look in particular at protein and at RNA simultaneously within small numbers of T cells. And I think that wraps up our overall webinar, and now we welcome questions. Thank you.
Thank you so much, Dr. Anderson and Dr. Heinerfeld, for your presentation. I'd like to now start the live Q&A portion of the webinar. If you have any questions, audience members, that you want to ask, please do so now. Just click on that green Q&A button at the lower left of the presentation window, type your questions into the box that appear on the screen, and click that send button. We'll answer as many as we have time for. So let's look at our questions now from our audience members. We have a great audience today, by the way. Are there issues of antibody interferences with the multiplex protein analysis?
Yeah. Hey, this is Doug, and I'll take this one. So this is something that we've looked at quite a bit. So with all of our assays that we develop, our multiplex protein assays, we do look at whether there's interference. And one of the ways we do that is if we stain with the antibody individually, or we stain with the antibody in the context of all the other markers in the panel, we can see whether the antibody behaves differently when you have other other antibodies in the mixture, which can happen. And if that does happen, it's not a problem necessarily, but we would take that antibody out, try a different clone, label it, and then test it again.
Great, thank you so much. Our next question is, how have you validated the oligo-tagged antibodies to ensure specificity and sensitivity, for example, the RNA?
Sure. So that similar kind of approaches. So with our, particularly the flow antibodies that we use in these assays that we talked about today, so we compare the results to flow cytometry data. I should say, and I think I mentioned before, all of our antibodies, we don't make antibodies. We buy them from vendors. We make that clone information available. Most of these antibodies are actually from BioLegend, so they're very well validated. We actually have a tech note that we wrote that describes the validation that we do that is on our website, so you can look at that for more detail. But again, we're starting with high-quality antibodies. We're adding a DNA tag. We're then making sure that we get the same responses biologically and otherwise that we would expect to see versus the gold standard, which is flow cytometry. So that really gets at the specificity of the antibody, as well as the ability to see the same sorts of changes, again, that we would see with flow cytometry.
Thank you, and I want to thank our audience members for our wonderful questions coming in. Do flow cytometry and nanostring antibodies block each other?
So that's also a good question, and it's certainly possible that they can. So this, I think, gets at really the 3D flow application where we're staying with the fluorescently labeled antibodies we need for sorting and the DNA labeled antibodies for quantitation on our platform. So we always check that. We certainly, recommend to our customers that they check it before they start a study, particularly if they're working with fluorescent antibodies that are different than what others have used. So very simple way to check that is to stain your cells, actually take your cells and split them into two tubes, stain them both with the fluorescent stained antibody, but in one of those tubes add our DNA labeled antibodies. Do a quick analytical cytometry experiment, and if you get good fluorescence for your key markers that you need to sort your cells, even in the presence of our antibodies, you're in good shape. If, on the other hand, adding our antibodies knocks down the signal, you know that those two clones are not compatible and that they're competing for binding. And in that case, either you still have enough signal to detect it and you can still sort it and you're fine, or if it knocks it down lower, you would then want to change the fluorescent clone that you're using and try one that binds to a different epitope on the protein.
Great, thank you, Dr. Heinerfeld. How do you normalize and analyze multi-omic data like this, and what controls are used?
Sorry, Karen, I keep jumping in and taking answers, but feel free to chime in. So we have all of our panels. So at the RNA level, we have a whole set of housekeeping genes that we use for normalization. In our software in Solver, we have a variety of tools that can be used to either use all the RNA or all the protein to normalize or using subsets of those. So it really depends on the study. So at the protein level, it's common that we will normalize based on what we call the geomean, that's sort of like the average of all of the proteins, and use that to normalize across the different samples. However, if you have a marker and you look at the data that you know doesn't vary much, such as CD45, you can just select that marker and normalize based on that. So the software lets you do pretty much anything you want, and it really comes down to the biology. We do also have some negative control antibodies that you don't normalize with, but are quite important to understand what sort of background staining you have in your experiment.
And I'll also chime in on this. There's also level of not just normalization, but comparison for controls that are used regarding biologic controls. So there's certainly the opportunity to do different control populations. So we've been looking at tetramer positive versus negative CD8 positive cells or CD4 positive cells. That can be done. You can look at stimulated versus unstimulated. Luckily, a number of these populations have been looked at in fairly deep molecular profiling studies, so that we have an expectation about the levels of T-cell exhaustion, for example, and the different levels of co-stimulatory or co-inhibitory biomarkers that are expected within these individual cellular subsets. So those are also built-in controls that can be used readily for on this platform.
Great, thank you, Dr. Anderson, and thank you, Dr. Heinerfeld. Here's our next question, and again, I want to thank audience members. We have some wonderful active participation today. Did you analyze nanostring data alongside the data from the flow sort?
So,
go ahead,
yeah,
Doug.
Karen. All right, I'll go ahead. Yeah. So we have done that. I mean, there was a heat map I showed in the talk where it showed essentially the sorted populations, which are obviously positive for the markers that we're interested in sorting on, also show up positive with the nanostring data. So we have those sorts of easy and obvious comparisons. We also have, of course, we look at like mean fluorescence intensity for the populations that we're sorting, partly to make sure that our antibodies are not interfering, as we talked about before. So I guess those are the two main places where we've really done sort of direct comparisons of the flow sorting data with the nanostring data. That said, you know, you've got both data sets in hand. In fact, there are some examples of a project that we're doing where there are antibodies that we don't have in our panel, but that they're interested in characterizing. So they've added those to the fluorescence cocktail. They're not sorting on those antibodies, but they're using those antibodies to phenotype their sorted populations. We happen to have RNAs for those molecules, so now they get the flow data, the fluorescence data for those markers that we don't have in the panel. They get our markers, which are in our panel, but they also get the RNA for those markers. So you have sort of, they're combining all those data sets in order to understand. Actually, it's another immune cell exhaustion project.
Wonderful, thank you. Our next question is, what cell types can be profiled with this assay?
Sure, so another great question. And so in terms of the method itself, right, the staining of the cells with the fluorescent antibodies, analyzing with the DNA barcodes, essentially any cell can be characterized. That said, there's a significant caveat to that, which is it does depend on the content of our assay. So currently our assay is very T-cell focused. So sorting and analyzing different populations of T cells, so it could be, again, you know, Tregs or antigen-specific T cells. Those sorts of samples, you're going to get a lot of great information using our RNA and protein content. Certainly, we are excited to develop more content to allow you to look at other types of cells. In fact, you know, if you have cell populations that you think this would be a great fit for, I mean, you can see on your screen there's the 3dflow@nanostring.com. We would love to hear from you so that we can understand what's the best content to develop.
Great, thank you. Our next question comes from across the country. How much diversity is present in the antigen-specific populations you isolate?
Well, it depends on how you measure diversity. I mean, there's certainly the antigen-specific diversity and the, for example, the T-cell receptor diversity can be quite high. Whether or not there is more embedded subtle differences in diversity in terms of the different levels of antigen-specific exhaustion, for example, really remains to be seen. I think at this point we've really only been looking at one population so far. We've been looking at Flu-M1 specific T cells, so I think that it remains to be seen how much individual diversity both between antigen-specific T cells and then within an antigen-specific T cell populations we can really look at. I think one of the major limitations, especially if you're looking at unstimulated populations, is precursor frequency. For that, you'll need, if you start looking at subpopulations within those cells, you're looking at a very limited number of cell populations. And that will be one of the limitations of being able to drill down to even smaller numbers of subpopulations of antigen-specific T cells.
Thank you, and thank you both for answering all of these wonderful questions coming in from our audience. We have time for a few more questions. Here's our next one. How long does it take to run an assay?
Well, I think that depends on, again, the precursor cell frequencies. The primary limitation in the time is the time that required for doing the flow sorting. And it's actually quite similar to what we would normally be doing for flow sorting. You have to prepare the cells. They have to be undergo antibody binding and all of the pre-analytic preparation that is required prior to the flow sorting. But the primary amount of time really ends up being in the time spent on the flow sorter to be able to collect those antigen-specific cells. The amount of manipulation after sorting is relatively limited, and then it takes additional time just to do the nanostring assay as well. But it is fairly comparable up until the time that we're running on the nanostring machine. It's relatively comparable to doing routine flow sorting for low-frequency antigen-specific T cells, at least that we've been doing.
Thank you. Thank you, Dr. Anderson.
Yeah.
Dr. Heinenfeld.
I can add,
I'll add
go
ahead.
A little bit. Yeah, I was just going to say that, so one of the strategies that's become really common, I think Karen mentioned this earlier, is doing a negative selection prior to staining so that you can reduce the flow time, the amount of time on the sorter. So that's one piece. But from start to finish, it is 2 days. So you're staining and sorting one day. You're immediately lysing those cells after sorting. You then take that lysed material, which again contains the protein tags and the RNA, hybridizing that directly to our barcodes. It's an overnight hybridization at 65 degrees. The next morning, it's loaded on the nCounter platform, and that evening you have your data. And to just take that raw data and import it into InSolver, generate do the normalization, and just get a sense of the results. Oftentimes, people are doing that the same day, so the end of the second day, if not the beginning of the third day. So it's very quick.
And is this workflow compatible with fixed and permeabilized cells for intracellular protein analysis?
Yeah, that's a great question, and one that we get asked a lot. As I pointed out there, we do have an intracellular protein panel, and you can add custom content to that. We've done a little bit of experiments with that. I will say that there are some, from the protein side, it's fairly straightforward, but the fixation and permeabilization does result in issues on the RNA side. So there are some modifications to the workflow that could be used, but certainly if that's something that anyone's interested in, we would love to hear from you. It's something that, again, we've done a little bit of experimentation on, But if someone's really excited about that approach, we could work to flesh that workflow out a little bit more.
Great, thank you. Here's an interesting question. Our audience member wants to know, I'm interested in very rare cell types. How many cells do I need to start with to have 5,000 for analysis?
Yeah, it's just a numbers game, right? So it really depends on, you know, the starting population, what the expected frequency is. You know, you should have some information about that expected frequency going into the study, either from analytical cytometry or from the literature. Once we know that frequency, then you have to ask the questions, do I need to do negative selection to enrich for populations before staining? Certainly there are limitations to how many cells you can stain, so reducing the cells if you're not interested in prior to staining is helpful. Yeah, so it really, you know, I guess it's simple math at the end of the day, just looking at the frequency, looking at the numbers of cells you have to start with, and then sorting until you get those 5,000 cells. I would say that, you know, 5,000 is a pretty ideal situation to get, of course, you get great protein counts even at lower amounts. RNA, you know, if they're stimulated T cells, or then even less than 5,000, we still get very good data. But certainly as you start looking at those unstimulated T cells that don't have a lot of RNA, the closer you can get up to a 5,000, the better data you're going to get. The data is not totally lost if you have less cells, but you start to lose some sensitivity for what might be some interesting markers.
Thank you to both of you. We have time for 2 more questions from our audience members. Our next one is, do antigen-specific T cells change during culturing, and how do you obtain a baseline reading for the in vivo biology?
Well, they certainly activate with culturing. They, I mean, the whole point of doing either short or long-term in vitro stimulation is to increase both the number and the activation status of antigen-specific T cells. It can dramatically increase your pre-test frequency of these cells, and so can improve the ability to do these in-flow sorting. But it also can change the intracellular cytokine levels, the amount of granzyme and perforin. It can change a number of different biomarkers of those cells as they get activated in vitro. So ideally, they would be tested prior to activation status, but there it really depends on the numbers, and if the target is to try to obtain 5,000 antigen-specific T cells, depending on the numbers and frequencies you're starting with, will impact, obviously, the strategic design of the experiment. And I think that that's one of the challenges, and I think that's also something we have the opportunity to be able to measure in a robust way, to look at what those changes are under different types of in vitro culturing and conditions and systems.
Thank you so much, Dr. Anderson. Our last question that we can do for today's webinar comes from an audience member. How does this compare to other RNA plus protein technologies?
Karen, you want me to take a stab at that?
I'll let you go first, yes.
Okay, but you can chime in from your experimental perspective. Right, I guess the biggest difference is really that it's integrated, that it's that same small population of cells that you're getting both the RNA and protein data, and the counts are digital, right? So it's really simple data to analyze. So the other approaches that people oftentimes use is, as I was saying before, high parameter flow cytometry or CyTOF analysis, and that's going to really allow you to phenotype and functionally characterize your cells, but it's going to be disconnected from the RNA data, and we've seen the advantage of having both RNA and protein. So for the RNA, again, you're going to sort the cells. People often sort them into a lysis buffer. Then for sort of non-nanostring methods, you then have to purify the RNA, generate cDNA, and then go into PCR, library construction, and then data analysis. whereas with the nanostring approach, we just lyse them and hybridize them to the probes, and you have really simple data the next day. So I guess that would be my sort of nanostring-centric comment on that. Karen, I don't know if you have some other perspectives.
Well, I think there's been a lot of effort in particular to develop and validate RNA-seq analysis, for example, on as low as single-cell work. And I think that much of that has been developed, and there have been a number of really outstanding papers on this. One of the challenges is that if you're going down to very few cells or even to single-cell work, it starts to become both cost ineffective and somewhat computationally unwieldy. And for many of the biomarkers that we're interested in, we don't necessarily need a full profile of every RNA biomolecule, but rather a targeted set, in particular to look at levels of T-cell exhaustion or levels of activation or T-cell activation or checkpoint inhibitor or checkpoint expression, et cetera. And so you don't necessarily have to look at the entire RNA profile, for example. And so I think that is one of the advantages, in that you're getting quantitative levels that you can compare multiple different subfractions of cells fairly rapidly, and you can get data fairly quickly, and it doesn't require sort of deep bioinformatic analysis. And I think that's one thing that makes it much easier to do a higher throughput analysis of relatively rare populations. Anyway, so I think there are a number of different technologies that are out there, but at least this allows you to be able to look at both the RNA, at least for a targeted set of RNA biomolecules as well as protein biomolecules.
Dr. Anderson, Dr. Heinerfeld, thank you so much for your presentation today. I'd also like to thank LabRoots and our sponsor, NanoString Technologies, for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through January 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thanks for joining us. We hope to see you again soon. Bye-bye.